All right. Hey, everyone. So my name is Jared Baldona, and I'll be talking about how to estimate parental effects using polygenic scores. So this is based on a model that I've worked on with Matt Keller and Yonggong Kim. And I've split this into two parts so that in this video, I'll be talking about some of the basics of the model before getting into its extensions in the next video. So let me turn off my video. Great. And I'll get started. So I think it's safe to say that we've all had the experience where we meet a friend's or a significant other's parents, and we think to ourselves, okay, yeah, that explains a lot. That makes sense. And that's an understandable reaction because from hobbies to level of extroversion and education, even things like TV show preferences and mannerisms, there are just so many ways that parents and offspring resemble one another, which begs the question, why? What is the underlying cause of all this parent-offspring similarity? To answer that, we can use education as an example, given that there is a large heap of evidence demonstrating that highly educated parents tend to also have highly educated offspring. So if we take the parent and offspring education phenotypes here, we can pinpoint a couple of different explanations for why they might be similar to one another. The first and perhaps most obvious of which is just shared genetic effects. So the parent has a strong genetic predisposition for higher education, which they transmit to their child, giving their child that same genetic predisposition. Easy enough. However, we can also imagine an alternative cause of parent-offspring similarity in which being highly educated itself impacts how parents raise their child. So for example, a highly educated parent might be more likely to encourage a positive attitude towards schoolwork or to read to their child before bed. Maybe they just happen to have more books lying around the house. All these different behaviors that collectively could potentially influence the offspring's education level down the line. So this effect of a parental phenotype on an offspring phenotype is called vertical or sometimes cultural transmission. And it's mediated by what's known as the offspring's familial environment. This is the environment where the offspring is raised, and it's created and shaped, at least in part, by their parents. So vertical transmission is an important and often overlooked source of trait variation in humans. And because the occurrence of vertical transmission will lead to traits running in families, it has been difficult for researchers to distinguish this vertical transmission path here from this shared genetic one that I showed a second ago. And it's especially difficult when those two co-occur, which will happen anytime that the trait being vertically transmitted is heritable, which, as you can imagine, is like almost all of the time. So as you can see, the same genes that are directly influencing the offspring phenotype are also influencing the parental phenotype, which in turn influences the familial environment. This means that children who inherit genes that predispose them to a given trait are also more likely to inherit a familial environment that predisposes them to that same trait. So when there is vertical transmission, these two things will typically not be independent of one another, but instead they will covary, with the covariance between the genes and familial environment being known as passive gene environment covariance, or more recently, genetic nurture. So like vertical transmission, the magnitude of genetic nurture reveals a lot of crucial information about the relative importance of parenting in shaping who children go on to become. It also tells us about the mechanisms underlying that process, which are definitely of interest. So for that reason, I think genetic nurture and vertical transmission are both really worthwhile things for behavioral geneticists to be looking at. But that said, even for people who are not interested in the study of parental effects themselves, vertical transmission and genetic nurture still have a number of really important consequences for BG researchers. For instance, the occurrence of vertical transmission means that the estimates of SNP effect sizes from GWAS will not only be capturing the actual direct causal effect that that SNP had on the person's trait, but also the effect that that SNP had on the person's parent which in turn indirectly influenced the individual's trait via the familial environment. So as a result, SNP effect sizes from GWAS will be upwardly biased to the extent that vertical transmission is unaccounted for. And meanwhile, for those of you who do twin research, vertical transmission will increase MZ and DZ twin covariances to the same extent, which will upwardly bias estimates of shared environment in twin ACE models. And less intuitively, I think, it will also upwardly bias additive genetic effects in twin AE and ADE models. So suffice to say, vertical transmission is not only a very important phenomenon in its own right, but it also has the potential to bias results across a variety of study designs, making it a really important construct for us to be accurately and reliably estimating. So to that end, a number of recent papers, most notably this paper in the bottom right from Kong et al., published in Science, have implemented a pretty novel approach. So instead of just looking at the genetic factors that are shared between parents and offspring, these ones in yellow here, that is the alleles that parents transmit to their children, these researchers have also looked at the set of alleles that parents have but do not transmit to their offspring. So that is the portion of the parental genomes that they do not share with their child. So like the transmitted alleles, the non-transmitted alleles still impact the parental phenotype, and thus they can still influence the offspring phenotype via vertical transmission. However, unlike the transmitted alleles, this is the only impact that the non-transmitted alleles can have on the offspring trait. Without this familial environment and this vertical transmission path here, there's no connection between the non-transmitted alleles and the offspring phenotype. They're totally independent of one another, with some exceptions that I won't get into here. So this means that by essentially subtracting these paths in green from these paths in red, we can get an estimate of this direct genetic effect here that is unconfounded by genetic nurture and vertical transmission. And we can use that direct genetic effect estimate to in turn obtain an unbiased estimate of vertical transmission and genetic nurture. So recently, Matt Youngong and I took this logic and instantiated it into a series of causal models that we've called SEM-PGS. So SEM-PGS has a number of advantages over previous approaches. The first of which is that using only polygenic scores, it can obtain unbiased estimates of heritability, vertical transmission, and genetic nurture. And it can do so even when the PGSs being used are not very predictive, which is great because most PGSs to date, if not all, explain a pretty small fraction of trait heritability. So in addition, they can also account for various types of assortative mating at both equilibrium and disequilibrium. And 3, if data are missing at random, then our model is not biased by missingness. So it doesn't require complete trios. If your sample just has father-child or mother-child pairs, that works too. You just need a larger sample size to achieve equivalent power. So 4, as I hope to demonstrate in these videos, the models are very easy to use. They're all structural equation modeling based, so they rely on generally pretty easy math. And 5, they allow for a lot of extensions to best fit the trait and data being worked with, again, by virtue of being structural equation models. So before getting into the full model, we can start here. So we have the paternal genes and phenotype on the left. Note the P subscripts for each one. And we have the maternal genes and phenotype on the right with these M subscripts. So we have these 2 Ys representing both the father and the mother's phenotypes, as well as these NTs representing the PGSs made from the non-transmitted portions of the parental genotypes, and these Ts representing PGSs made from the transmitted portions of the parental genotypes. And because these 2 transmitted PGSs are shared between the parents and offspring, that means that they have the same effect on the offspring phenotype that they do on the parents. So note the O for offspring here with this trait. So just as the Ts and NTs sum up to be each parent's full PGS, TP and TM sum up to be the offspring's full PGS. Now, if we take this diagram and throw on some variances and path coefficients, as well as some residuals for each phenotype, we now have a complete structural equation model for each person's direct genetic effects. So pretty much just illustrating this little diagram that I showed a couple seconds ago. So to go through these different coefficients one by one, this model contains delta, which is the magnitude of the direct genetic effect, and it serves as a reflection of the PGS's R squared. So note that all of these different haplotypic path coefficients have a delta. So we're assuming here that the PGSs are equally predictive in the parental and offspring generations. So in addition to these deltas, we also have K for each haplotype's variance. And K is just a constant that we supply to the model based on how and when PGS is scaled. So Ks are not estimated. And for the purposes of this video and the next one, you can really just think of K as being equal to 1/2. Finally, we have these VEs, which are the proportion of variance due to the individual's unique environments. So these are capturing the remaining variance that isn't covered elsewhere in our model. So while this is a complete structural equation model, it is by itself not very informative. It's pretty much just telling us how predictive the PGS is, and it's not telling us anything about vertical transmission or assortative mating. However, if we add these 3 latent F variables representing each person's familial environment, that will allow us to estimate way more information. So like I said, these Fs represent each person's familial environment. That is the environment in which they were raised. So we have the offspring's rearing environment, which you can see is shaped by the parental phenotypes. And these parental phenotypes are in turn influenced by the environments in which they themselves were raised. So we got the mother's familial environment and the father's here. So by modeling each person's familial environment, we can estimate W, which is the genetic nurture covariance. So that is the covariance between each person's haplotypes and their familial environment. And these Ws are accounting for the piece of these PGSs that is actually tagging environmental effects and vice versa. So in addition to these Ws, we also have these little f's, which as you can see are the impact of the parental phenotypes on the offspring phenotypes mediated by the familial environment. That is, these are the vertical transmission effects. And finally, VF on these two ends, which is the proportion of phenotypic variance due to vertical transmission. So that is the full portion of phenotypic variance that can be explained by parenting and parental influences rather than genetic or other environmental effects. And because this is a proportion of variance, it's on the same scale as things like heritability and VC and VE from twin family studies, allowing for comparisons across methods. So this is what we kind of arbitrarily referred to as model 0 in our paper. And it can account for and estimate vertical transmission, but not assortative mating. But nonetheless, I think it illustrates a lot of the concepts that are common to all of our models. For instance, by comparing variances and covariances between these 5 observed pieces of information, these 5 observed variables, if we compare the variances and covariances between them with their algebraic expectations that we get from path tracing, we can estimate all of the parameters shown in the model. So these deltas, these Fs, these Ws, these VFs. To give an example of how that works, I'll show how we can obtain the algebraic expectation for the covariance between the offspring phenotype and the non-transmitted paternal PGS. So to do this, we just need to trace all of the paths between YO and NTP that abide by a specific set of path tracing rules, which are shown here. And I won't go through this because that is a lot of text, but feel free to pause the video and read through these if they're of interest. We also describe them in some detail in our paper if you really want to kind of get the nuances of these down. So for this covariance between these 2 variables, there are exactly 2 paths that meet all of those path tracing criteria. So once we've identified what those paths are, we can multiply the coefficients within each path and add that to the other paths. So for this path on the left, we get 1 times F times 1 times half W. And for this path on the right, we get 1 times F times delta times K. So if we add those two products together, that leaves us with half FW plus F delta K, which is the algebraic expectation for this covariance here between YO and NTP. So all in all, it's pretty straightforward and not super mathematically involved. It's really just like basic multiplication and addition, which is great. It's easy to use. And while I'm here, I'd also like to point out that you can see how both of these models are going through YP. And that makes intuitive sense because with no assortment, the only way that your father's non-transmitted alleles could be impacting your phenotype is through their impact on his phenotype, which in turn influences how you're raised and how you grow up. So that's another great thing about structural equation modeling and path diagrams. They allow you to visually see relationships between variables and to understand how the variables are acting on one another. So finally, I'd like to point out, which you may have already noticed, that there are some variables that are included for the parents, but not for the offspring. So for example, we have these W's in the parental generation, but we don't have W's for the offspring. Same thing for these VFs. So that doesn't mean that we can't estimate W and VF for the offspring or that those variables just don't exist in the offspring generation. But instead, excuse me, it just means that these values are implied rather than shown. So in other words, just as the W covariance between the parents' familial environments and their haplo-PGS is shown here, we can also get W in the offspring generation by path tracing from the offspring's familial environment to their two haplotypes. So if we were to include an actual, double-headed arrow, W arrow between these different variables, that would be redundant. We would be including the same thing twice. Same goes for if we included a VF variance right here. Okay, so that is model 0, and it's where I'll end for this video. So thank you all for listening, and I'll see you for part 2 when we discuss some of these model extensions. Thanks.